All right, so like I said, from the lungs down to the bladder, E. coli sequence type 131 is a pandemic clone. It's multi-drug resistant and it causes urinary tract infections. So a little bit about antibiotic resistance. It does have a global impact. I don't think anybody can argue with that. There was a paper published in the Lancet in 2022 that looked at just 1 year of antibiotic resistance uh, globally, and almost 5 million deaths were associated with antimicrobial resistance in 2019. Of those deaths, almost 1.3 million were directly attributed to the antimicrobial resistant organism. The highest death rate, as much you expect, was in sub-Saharan Africa, the lowest death rate in Australia, and the 6 leading pathogens, E. coli, Staphylococcus aureus, Klebsiella pneumoniae, Streptococcus pneumoniae, Acinetobacter baumannii, and Pseudomonas aeruginosa. I highlighted E. coli because it is at the top of the list. So the burden of UTIs, a lot of people think, oh, it's just a UTI. Well, it's not just a UTI. There's a global burden of 150 million people contract a UTI every year. 50% of women experience a UTI by their late 20s. 20 to 30% of those women experience recurrent UTIs, and 5% of those recurrent UTIs are chronic. 5% doesn't seem like very much until you do the math, and it's about 7.5 million people a year. So what about UTIs? They are a major medical issue. Patients suffering, loss of wages, a lot of women who have them have jobs that if they take it off, they don't get paid. So they have loss of wages, increased medical expenses, and 25% of the sepsis in hospitals are due to urosepsis. Of those 25%, 25% of those are in the sequence type 131 that I'm going to talk about is what's causing that urosepsis. And this is just a graphic showing that in uncomplicated bladder infections, the culprit is mainly E. coli, and even in complicated cystitis, it is still E. coli. So what are these extraintestinal pathogenic E. coli? Well, yes, the bulk of diagnosed UTIs, whether it's cystitis or pyelonephritis, they cause both healthcare-associated and community-acquired infections. The cause of UTIs, bloodstream infections, prostate, other non-intestinal sites. And a lot of these organisms, especially the 131, the E. coli, have multiple drug resistance. Beta-lactams, they carry extended spectrum beta-lactamases, plasma-encoded MC beta-lactamases, and carbapenemases, along with fluoroquinolone resistance, aminoglycoside resistance, and resistance to antimetabolites. The 5 sequence type complexes represent over 50% of the XPEC isolates. Sequence type 131 is the most prevalent XPEC clone globally. So just to show you a little bit of why or how they came to those data, here is a list on the x-axis here of all the different sequence types that are the most substantial in the world. And as you can see, sequence type 131 is found in North America, Europe, Asia, and Africa, where a lot of these, especially these in this area, are not globally found. And then according to this particular graph, you have UTIs, colonization in the gut, and bloodstream infections. Again, the winner here is sequence type 131. There was a study done for kids. Usually we look at urinary tract infections in adults, but there was a study in France that looked at febrile urinary tract infections and found again that the culprit was sequence type 131, but there's some data out there, which I'll share at the end, where sequence type 69 is becoming increasingly difficult for kids. And then sequence type 1193, which wasn't even around in 2015 or 2016, but now is starting to emerge as another significant sequence type. So what do we know about these bugs? The majority of the data distinguishing 131 strains has been with whole genome sequencing. And this methodology underscores specific genetic differences among and within these strains. But the problem is that there's no specific differences that can contribute to the success of the sequence type 131 pandemic clone. There are no predictive set of virulence genes they've tried that distinguishes 131s from other 131s, as well as the non-131 strains. And of the virulence genes that are the same, they're chromosomally encoded. So fimbriae and adhesion genes are involved in binding to the epithelial cells. I've highlighted this one because it's rather unique. It is a membrane-associated protease, which actually cleaves antimicrobial peptides. You don't see this in all strains, but I want to come back to this in some of our preliminary data. Few physiological differences have been elucidated, and of those studies have been done only in culture, not in animals. So to date, I can't find any data in the literature that can explain the global dominance of these sequence type 131s. So that moved us to our question, what is it about these organisms that made them globally dominant, but still no answers? It could be fitness. So when the bug infects the bladder, is that environment a fitness advantage, or is it really pathogenic and virulence advantage? Don't know. So here's our central hypothesis. Yes, it's a long one. The variability observed in the genomes among the strains of E. coli 131 reflects the ability of these organisms to cause kidney or bladder infections in a mouse UTI model. And those differences in the infectivity, excuse me, are linked to differences in metabolic pathways during the kidney and or bladder infection. So I have four people I want to thank right off the bat. This is Travis Beret, associate professor in our department. And if it wasn't for him talking me into working with animals and letting him do the IACUC, I wouldn't be having anything to talk about. And then I have three young women up here, two master's students, Rachel Cox and Holly. Rachel started these mouse infections for me. Holly did some animal sequence type 131s, but Courtney has really taken the ball and has done the most work with what I'm going to present to you now. And she drew this diagram. So what we have done is we've taken a couple of strains for preliminary data, well, more than a couple. We've run them through culture, all right, and then we get them RNA sequencer RNA, the transcriptome. We've also taken some organisms and put them through this UTI model for mice. And then instead of culturing them, we take urine over a time course, look at the CFUs or the colony forming units, or the number of bugs. Then we take the kidney out directly and send it for sequencing. And then we can do dual RNA-seq and know what the immune system of the mouse is doing and what the bacteria is expressing. So with our first preliminary data, we looked at a few isolates, some a laboratory strain, some that were non-131 and some that were. Whoops, oh shoot, now I'm not in the right, get rid of this. So when we looked at this, we had different sequence types. Here's the number of mice we infected. With this sequence type, we saw infections in 2 mice in the kidney, 1 in the bladder. In this 131, we didn't see any infection in the bladder or the kidney and very little in the urine. And this XQ12 is another 131, and we had almost 100% infectivity in the kidney. So we've got two 131s right off the bat that are not acting the same. So when we looked at the transcriptome of these three organisms, 2,900 genes were shared between the three, even though their whole genomes were approximately the same size. The one that went to the kidney the most had 43 unique genes. The one that didn't go to the kidney at all had 10 unique genes, and they were both sequence type 131s. This one is a non-sequence type 131, shared 9 genes with this guy and 6 genes with this one, and had 191 unique genes. So again, the 131s are more genetically similar, but not identical. So then Courtney did another huge study where she looked at a lot more mice, and to make a long story short, let's see, get this on here. To make a long story short, these are all 131s. You can see that 9 out of 10 went to the kidney in this particular organ, in these organisms, with high CFUs of 10 to the 7th, 10 to the 8th. But again, here's another 131 that went to the kidney twice, but had pretty good CFUs. But we get down here, this 131 didn't go to the kidney at all and had no CFUs. So, just to show you, we know what an infected kidney looks like. Here it is, nice and infected, and here's one that's nice and shiny. So when we looked at the RNA-seq data, so what we did was to compare this organism here is the one, I'll call that the 100% to the kidneys, and this one is 50% to the kidneys. When we looked at a subset of genes between the two organisms, we didn't see a lot of difference, which is what you would expect. But when we compared each organism to the kidney versus culture, which most people do this in culture, we saw a huge difference in the fold difference. So this is log 2. So actually the fold difference, we saw a downregulation of OMP-C and OMP-F. These are two porins in the outer membrane that allow antibiotics to get into the E. coli cell. OMP-N, if you look at this transcript in culture, it's non-existent. And yet in the kidney, it is overexpressed, well, let's see, about 10-fold. OMP-T is that membrane associated protease that cleaves those immunopeptides. It is also expressed higher in the kidney than what you would ever identify in culture. But there still has to be differences between these two. So what Courtney did was to make a volcano map, and even though these aren't as statistically significant as we would want them to be, we still are seeing differences. So in the robust infector, W15, we had 81 genes that were upregulated in this organism, but not this organism, and 102 genes that were downregulated in this robust guy compared to this one. So the take-home message is that sequence type 131 E. coli are a dominant pandemic strain. The humans are the main host, but there are spillover hosts, including dogs and cats. So when your cat or dog gets a urinary tract infection, it could be infected with one of these 131s. And in some studies, the literature suggests that we can see transfer between pet owners and their dogs or cats. You can see them in meat animals, swine, poultry, beef, and also in the environment. Hopefully, I've shown you with a little bit of preliminary data that we have that sequence type 131 E. coli are not all the same. And studies that we've done in the past and have published using culture, organisms from culture, we see differences in the expression of the extended spectrum beta-lactamase, or that cephalosporin resistance, as well as the porins, that membrane on the outside of the proteins on the outer membrane, we get differential expression of those as well. The types of virulence factors are not all the same, and the ability to infect the kidney or bladder are not all the same. So I put together, the next slide is for some collaboration that we could do with our infectious disease docs. And here they are. I think we need surveillance studies in Nebraska and Arizona, and actually a lot of other places, because the Europeans and the Australians, they seem to do more surveillance studies than the US. So my questions are, what is the proportion of sequence type 131 E. coli versus non-131 E. coli in Nebraska and Arizona? We have no idea. What's the urosepsis rate for hospitalized patients? We could probably figure that out. And what is the urosepsis rate for hospitalized pediatrics patients? And is it correlated with a particular sequence type? There were 2 studies in Mexico and France that associated pediatric hospitalization with sequence type 69 as well as sequence type 131. And then there was a study just recently from the University of Washington State in the Kaiser healthcare system. And this is interesting because everybody thinks that if you decrease the use of antibiotics, you will decrease the carriage. And that's not what happened in this case. There was a 3-fold drop in the prescriptions for ciprofloxacin, which is a fluoroquinolone, but the gut carriage of fluoroquinolone resistance was up from 14 to 20%. Does this suggest that the fitness of these organisms are already ingrained in the gut and it doesn't matter? There's no selective pressure for the antibiotics being used because they're already environmentally tuned to where they're living. And then the co-resistance to the 3rd generation cephalosporins also increased from 14 to 32%. And the last one is there a link between 131 and sequence type 1193 in urosepsis in Nebraska and or Arizona? Because in Florida, there was a little, a baby that was infected with sequence type 1193 and died of encephalitis. So on that grim note, I will share, I just want to say that even though not every student and every tech in the lab has worked on this project, it's all of their effort that has led us to be able to do these types of experiments. And then these people here were the ones that were most involved. So with that, I'll be quiet.
Thank you, Nancy. That's great. Go ahead, Renuga.
Oh, yeah. With your question about, that was really good about the urosepsis. I think we could easily design something because, as clinicians in the hospital, we see a lot of E. coli urosepsis. So I think if there's a way, the ID team, the Dr. Cavallarian team, and yourself, you could figure out who has urosepsis and to get those isolates, I think that would be a really good study.
If we could get the E. coli, I don't want the urine, I just want the E. coli.
E. coli, yeah.
But there's a nice multiplex that we can run that will give us an idea of the 131s and then what branch of 131s, what clade it might be. And so I just think, even if the antibiotic treatment doesn't change because of the resistance mechanism, knowing that you have a bug that is much more virulent, you can watch your patients for sepsis, because I'm not in the hospital, but it's my understanding that sepsis happens really fast. I mean, your patients are good and then your patients aren't good. And if you knew you were dealing with an organism that has a propensity to cause urosepsis more often than others, it might be helpful just to keep on that patient so that it could happen. I don't know.
Yeah, I think there is opportunity there, especially we call it asymptomatic bacteriuria. A lot of patients are just colonized, so we don't chase after that. But we do have a lot of patients who become septic and septic shock with
Yeah,
coli sepsis. So I do think there's an opportunity to even just see what are the isolates that are causing severe disease due to E. coli, and is it these strains? Even
yeah.
just to first that question, we probably should have a brainstorming meeting, but the main person is Dr. Cavallari. We need him.
Well, we can talk to Dr. Cavallari. He lives in my building. I can go up and, just harass him upstairs at night.
I've got a little question. I know that, as we pursue the purity of the science, we like isolated models, and we, are basically selecting out for the resistant bacteria, because when we treat with antibiotics, the sensitive ones die out. But do we know much about the ability of different strains of E. coli to sort of coexist? That is, if you inoculated several strains together in a culture, would one emerge as a dominant over the other? And might there be other sort of, culture environments that would favor or disfavor particular strains? Now, this may be a little tangential. I'm not sure if it's at all practical, but I wonder if there is somehow we could find factors that promote a less virulent strain to help choke out the more virulent or more resistant strains, other than exposure to antibiotics.
Well, I think there are multiple strains that are colonized in the gut, and you can do those competition experiments once we figure out, like, okay, what happens just because we can collect urine over time, and we could look at the organisms over time to see which ones, and then when we do that, we also do susceptibility so we can figure out which organism is which. And we could make strains, but we could also take that strain that couldn't go to the kidney at all and mix it in different proportions with the one that did, and then see how long, which ones lasted longer in the urine and in the bladder. Because right now I don't have any data. We have bladders that we've frozen, but we don't, this was all preliminary pilot stuff. Mm-hmm. I need a little more money to do this, but we could get bladder tissue. And then, because another question is, if they're in the bladder, do they always go to the kidney? Or are there some that are just happy in the bladder and they stay in the bladder, and then you can get rid of them because you can get a high dose of antibiotic in the bladder? Or is there something special about them traveling up the ureters up to the kidney? And then another question I have is, because we see a downregulation of those porins in the kidney, does that influence the ability of an antibiotic to do its job, right? And we can do those experiments and ask that question, because if we have some isolates that don't downregulate for some reason, and since I've only got a couple to look at right now, I'm hoping to expand this. And then we can, I think we can answer those, we can start to answer those questions.
Nancy, this is, I just love, I love antibiotics. I love antimicrobial resistance. But it's fascinating work. And as you mentioned in the very beginning, it's of a scale that's really important. Not all resistance, obviously, and all these issues are important, but this affects so many more people than most of the other issues. So one thought is, in addition to, is get urology involved. So, you and Renuga, so you, ID, Pharm, and urology, I think might be really important, because I think there's a lot of urologic infections, urosepsis, et cetera, that doesn't necessarily get an ID consult, and working closely together with them to make sure those all get captured If you need, obviously, any kind of bladder tissue, they're your folks, right? And, one other, point to sort of throw out there, and I don't know if you've ever studied this, are Staghorn calculi of the kidney. When you talk about 5% of people who have chronic urinary tract infections, Staghorn calculi of the kidney are actually very different than others. They're soft, they grow slowly, they're silent, but they happen primarily in women who have recurrent urinary tract infections. And they grow over time, and they can sometimes take up half to the whole kidney, and they're found incidentally. But they might be a pretty unique subgroup.
The other thing we would like to do, and obviously there's a lot to do, but some of these E. coli can get into the epithelial cells. They're called IBCs. And so then the question becomes, what allows a bacteria to make an IBC versus others that don't? And does that recurrent infection come from these maybe hidden or stealth E. coli in a cell? And what allows them to do that? I don't know. So
okay, I'm gonna, this is really interesting, lots of interest. I'm really wondering whether John, you or other people at Phoenix might want to be part of the conversation. I have another meeting to pop to. I'll just officially end this wonderful research brainstorming. Thank you very much to our presenters for doing such great work and sharing it with us. And if you want to continue to talk, I think we can leave the Zoom session open. And we will see you next time.
thank you.
Thank you.
I've got a meeting, so I'm going to hop, but please continue talking.
I'm going to cut out too, but just say I'm a little bit far removed. Earlier in my career, I had a lot of interest in surgical infections, and I was probably more, familiar with the latest than I am now. But I'm interested in the process of science, always willing to discuss these things and enjoy the intellectual stimulation of these projects, even though I'm not directly involved. So I'll do whatever I can to help advance the.
Well, if you have any interaction with your ID colleagues, I'd be really, really interested in not just E. coli, but I think Phoenix will have a different set of resistance mechanisms than what we see in the gram negatives in Nebraska.
Yeah.
And if they ever want to share some isolates with me, I'm always building my repository.
Well, I'm here in Omaha. I'm on the Nebraska side. Yeah, but no, I know what you mean. It's good to, geographic differences can emerge and show patterns. And I'm always been fascinated by, the three components here. When I used to talk about infection and antibiotics, we talk about host factors, the organism, and then the drug. And I think we sometimes over-focus on the drug and forget to pay enough attention to the organism and the host. And so if there are ways that, we can map that out, like you're doing with the characteristics of these various strains and the genomics, maybe we can take the pressure off the antibiotics of having to do all the work.
I think that the more you know about the bug, the better off you are to treat the bug.
Yeah.
And the other thing I like about this model and taking it right out of the kidney is we haven't had a chance. I'm not an immunologist, but there are some gene expression from the mouse immunological side that I think we can use as well. So that'll be, I'm not an immunologist and I've barely got into this. So the urinary tract, I'm learning. That's what we do.
All right. Well, thank you very much. I gotta bail.
Okay, bye.
Bye.
Hi, Dr. Hanson.
Hi there.
It's been a while, but thank you. That was a really wonderful talk. Quick question. Did I read it correctly? If you do those studies for 131 in broth, you would get completely different gene expression results compared to in the
Not maybe completely, but a different intensity.
right.
Okay, so something in culture that may not be statistically significant is statistically significant in the kidney.
Right. And if you remember, I'm a drug discovery person, and that's the kind of thing that just makes my ears go boop.
Yeah.
It's doing something virulence wise. You've got to go target those things and see if you can control some of those genes, and if that really helps. I'm sure you already know, but there are libraries, transposon mutant libraries available for E. coli, which are not 131 relevant. I'm sure I'll have to take a look into it, but immediately I thought about
The laboratory strains that we've used, I don't know if you're talking about the Kyle collection or not, but
the one from Yale. I can't remember.
yeah, the Kyle collection. We've used the porin knockouts, single porin, double knockout, and the laboratory strain. They don't even stay in the urine for more than a day or 2
Yeah,
days.
so it's just not relevant.
Yeah, they're not really relevant. We use them to show that they're not relevant as a control, but
Yeah, I'm assuming you're not going in that direction, are you?
in the
Vaccine.
laboratory,
Yeah, no, I mean the 131 strain, if you the one that's most relevant, do you think, are you thinking of going down generating transposon mutants, looking at it that
well, in the future, we're gonna not transposon. I'd like to do CRISPR.
Oh, cool.
Yeah, we've got
One
some
gene at a
plasmids in the lab now. And the problem is with resistant organisms is when they make these plasmids for manipulating things, they use resistance selection markers that our bugs are all resistant to.
Right.
So before we can even start that process, we have to engineer the vector that we have to replace the resistance marker. And we have to use like a carbapenemase or something because otherwise we wouldn't be able to select it.
Right. Yeah. No, that.
So we have lots of issues that people don't realize that you want to work. And if you don't work with the bug, the clinical bug, you're not going to learn anything.
Right. Yeah. No, I agree completely. Well, we've got a ton of thoughts. Maybe I'll ask you for a meeting, maybe sometime late next semester or something like that, if that's okay with you.
That sounds great. I'm off to Fort Collins before the snow starts to see my grandson and trick or treat with him. So I've got to go as well. So thank you, Brianna.
Thank
Appreciate
you.
that. I'm sorry, I don't know why it worked when we did it.
Yeah, I don't know. It's all right. It works out. We're all good.
The talk
Not
was
known for my
great.
technical savvy. So okay, guys, thanks a lot.
Thank you.
Happy Halloween.
Bye.
You too.